Hi, everybody. This is my dog, Tiny. Uh, and we're in my backyard today, talking about breathing. So this is actually probably going to be the last of these lectures from the coronavirus series that I've been giving to you guys. Because urinalysis is next week's lab, and normally that does not include an introductory lecture. I'm not quite sure what it's going to be yet, so we'll see. But today's lab is all about breathing, or this week's lab is all about breathing. In other words, respiration. Not to be confused with cellular respiration, the thing that your mitochondria do in your cells. So you're going to be doing a couple of exercises this week around the process of breathing. Of course, the human respiratory system centers around the lungs, but also includes the bits of your body that get the air into those lungs. So the nose and nasal cavity, the pharynx, the larynx, of course, which is used to help you talk. And then as the air passes through the upper part of your respiratory system, it will go into the trachea, which branches into two bronchi, which lead into the lungs. Of course, a lot of this upper part of your respiratory tract is essentially dead space because it doesn't do anything for you as far as gas exchange goes, because that all happens in the capillaries, right at the end of these bronchioles, in these little structures that look like bunches of grapes that are called alveoli. So your lungs are basically spongy organs in your thoracic cavity, and they're basically designed with surface area in mind. So the idea is that the millions of alveoli that are terminal to your bronchioles, these little bunches of grapes, again, as you can see highlighted in this circle here, have an amazing surface area to allow the air that is, the oxygen that is in the air to diffuse into your blood, and the CO2 that's in your blood to diffuse back into the air. Because basically gas exchange is the goal of your lungs, when you get to these little alveoli, you notice that they have a very intimate, an intimate relationship with your blood supply. And this makes sense, right? Because basically you're trying to maximize the amount of blood that is getting as close as possible to the air itself. They're also surrounded by these elastic fibers to help stop them collapsing, because they have to stay inflated in order to be filled with air for efficient gas exchange. Notice that you can separate the lungs into sort of two general areas, shown by this dashed line. To the left of this is called the conducting zone, and that's essentially everything, all of your respiratory system that is conducting air down into your lungs. And then where the business actually takes place is in the respiratory zone, which is where the alveoli sit. So the pulmonary capillaries are where the gas exchange is going to take place, and we're going to zoom in to that structure for just a second to look at it in a little bit more detail. So what you see if you look at this in more detail is basically this is one alveolar sac. So this is one of those little grapes in that bunch of grapes. You can see they're cut out above the zoom in. And you can see the three types of alveolar cells. There's type 1 cells, which essentially are the dog tickling my foot. These very flat and thin cells. If you take a cross section through them, you can see how thin they are, and you can see that there's essentially a very thin membrane known as the respiratory membrane that separates the air in the alveolus from the blood, just the other side of these cells. So we have the capillary endothelial cell, a very thin basal lamina, basically connective tissue, and then the very thin type 1 alveolar cell. Type 2 alveolar cells are these cuboid cells. They basically secrete mucus to coat the inside of those alveoli and allow this surfactant to prevent your alveoli from collapsing. And the third type are these crazy looking alveolar macrophages, which are basically part of your immune system that crawl around the inside of those. They really do move around the inside of those alveolar sacs, getting rid of bacteria, anything that you shouldn't be inhaling, like particulate matter can be ingested by those macrophages and destroyed. So this respiratory membrane is where gas exchange takes place. And if I just put in the little gas exchange insert, you see what's happening. Oxygen is obviously moving into your blood. Carbon dioxide is moving out of your blood. And if you remember back to the very first lab, this is just occurring down concentration gradients, right? So the venous blood that, well, I guess it's blood is centered around your body by your systemic blood circuit, right? By the heart. Your muscles use, and your other cells use up the oxygen for cellular respiration in mitochondria, the manufacture of ATP, essentially. And that decreases the concentration of oxygen in your blood. The venous blood supply gets it back to the heart, which pumps it around the pulmonary circuit for oxygenation. So the arteries that take blood away from your heart towards your lungs contain deoxygenated blood. Because it's got a lower concentration of oxygen than the surrounding air, oxygen diffuses into the blood. And of course, the opposite concentration gradient is driving the movement of carbon dioxide. You have a high amount of carbon dioxide as the blood is returned to the lungs because it's collected that as a waste product from your mitochondria, essentially. And because it's at a higher concentration than the CO2 in the air, it diffuses out and is removed from your blood supply. Isn't that right, Tiny? My dog's called Tiny. So muscles of breathing. So how do you actually get air into and out of the lungs? The main muscle that causes that is the diaphragm. It's kind of shaped like a parachute, sits right underneath your rib cage. When it is relaxed, it is the shape of a parachute. It's kind of like a concave shape. And when you want to breathe in and you want to get air into your lungs, you contract your diaphragm, which flattens the muscle out. And as you can see on the right, this little GIF, when it becomes flat, it increases the volume in your thoracic cavity, which decreases the pressure in that cavity, which causes air to rush in, because you're basically decreasing the pressure in your lungs to below air pressure. When you breathe out, you essentially just relax your diaphragm. It goes back to its concave shape because your lungs are elastic. And that decrease in volume forces air out because you're increasing pressure by decreasing volume. And you'll look at that a little bit in your assignments today. It's actually kind of interesting. If you look at this real image of a human being taking a deep breath, you actually see how much your internal organs move around. The heart is in the middle here. You can see it beating. But right here, you can see the liver. And you can see when this person takes a deep breath in, they are contracting that diaphragm, which is flattening out. When that happens, it actually pushes down on the liver and kind of squishes it down, as you can see in this diagram, in this GIF. So wait for it. There it goes. It's getting squished all the way down, which just is kind of cool because it shows you how, much your internal organs can move around. When you're breathing at rest, which is called your tidal breathing, your diaphragm does most of that. When you need to breathe a lot more, if you've been exercising or something, then you have different muscles that are used. So these are called accessory muscles, and they're muscles like the intercostal muscles shown here in between your ribs. You can also use your abdominal muscles and some of your back muscles to help you expand the volume of your chest when you need to. But normally when you're sitting there at rest, you shouldn't be using your accessory muscles. You can tell when somebody is using their accessory muscles because their shoulders start moving up and down. And if you see someone doing that just for quiet respiration, it means they probably have some problem with their lungs in some way. So if you see someone with emphysema or some other chronic obstructive pulmonary disease, then even at rest, they're like, shoulders are moving because they're engaging their accessory muscles. If you do have some kind of problem with your lungs, then you tend to see some of the following symptoms. Nasal flaring, because you're trying to get as much air into your body as possible. Cyanosis, your lips might turn blue. Also, the beds of your fingernails can turn blue. Your chest can feel tight because you're using those accessory muscles. By the way, that's another reason why your chest feels tight if you're suffering from anxiety, because if you're anxious, you activate your sympathetic nervous system, your fight or flight response. And when you activate that, you tend to up your respiration rate because you're meant to be preparing for some kind of, forceful altercation or some, emergency situation. And when you are doing too much breathing, it causes chest tightness. So these are all examples of inadequate breathing. Sorry, I'm laughing because I'm just getting dizzy, which is one of the symptoms down here because I just did too much heavy breathing. So to see if somebody's lungs are working okay, you can do pulmonary function tests or PFTs. And basically, you can set up a situation like this gentleman down here, where you breathe through a tube. And this thing that he's breathing into is called a spirometer. And you'll be looking at some spirometry data this week in your assignment. This guy is actually doing it wrong because one thing that you're meant to do with a spirometer is have a nose clip on, because you're meant to just breathe in and out through your mouth to measure, total breathing in and out of your lungs. You're not meant to breathe in your nose at the same time. But this kind of device is used to measure breathing rates and capacities of your lung and things like that. And you're going to look at some of this data today. So I just want you to look at this graph for a minute and see what kind of volumes that you can measure using a spirometer. So what you are seeing when you look at a graph like this, and you're going to see some example data that you're going to analyze this week, is that on the y-axis here is the volume measured in liters of air that's going in and out. And on the x-axis along the bottom is just time. So when you breathe in, the spirometer measures the volume of air that you're breathing in, and that is shown on the graph as an uptick on the volume scale over time. And when you breathe out, the graph will go down. So when you're looking at the data that you're going to look at this week, that's what you're going to observe. Now, you can ask individuals to do certain things when they're hooked up to a spirometer to see what their kind of maximal capacity of their lungs are and things like that. And the way that you can do that is, so if you follow this purple line, this up and down is called tidal volume, as shown here. And that is just your breathing at rest. And then what this individual is doing is a pulmonary function test where they've been asked to breathe in as hard as they can, which measures as a big uptick in volume. And then they're asked to expire all of that air until they cannot expire any more air. And so that it goes down until you see how much they can expire. This volume is known as the vital capacity, which is basically as much as you can possibly breathe in and out. Of course, it's impossible to completely empty your lungs, You can't completely squeeze all the air out of them. And so notice that the vital capacity here is smaller than the total lung capacity. There is what is called a residual volume, which is the volume of air that you can never breathe out. So you can do lots of tests like this to see how functional someone's lungs are. One of the ones that you'll look at today is forced expiratory volume, I think it's called, which is basically as much volume as you can blow out in 1 second. And of course, the longer you take to do that, the worse off your lungs are functioning. And that's it. So enjoy your assignments this week. There are 2. Please do them in the order in which they are indicated on the website. And I hope that they are fun. As always, please don't hesitate to contact me if you run into problems. All right, if I don't lecture to you guys next week, thank you for being in my class, such that it is. I am bummed that I didn't get to do a proper class with you guys because it was fun. But hopefully I'll see you around campus. Unless you're graduating, then maybe I'll see you at graduation, whenever that may be. All right, guys, take it easy.